Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar on large-scale genome editing of primary human NK cells, a Quest Award manufacturer of clinically relevant CAR NK cells. I'm Elisa Manzotti, founder of BioInsights, and joining me today is Dr. Deepak Kumar, who will present data demonstrating successful engineering of CAR NK cells at clinical scale. Following his presentation today, there'll be a live Q&A, and as always, do feel free to pose your questions to our presenter. Just use the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. I'd also like to draw your attention to the Resources tab on the right, where you can find out more information on the topics that we're going to be covering today. But first, I'd like to introduce our presenter. Dr. Deepak Kumar is a scientist at Thermo Fisher Scientific, where he focuses on NK cell gene editing using viral and non-viral approaches to support cell and gene therapy workflow applications. Deepak received his PhD in molecular biology and genetic engineering from the Institute of Life Sciences in India. So without further ado, I'll hand over to Deepak today to kick off our presentation.
Hello, everyone. Thank you for joining the webinar today. And today I will be discussing about the large-scale genome editing of primary human NK cell. So overall, today's agenda will be, we'll talk about overview about cell therapy manufacturing, NK cell therapy, a small-scale electroporation optimization for NK cell genome editing, NK cell genome editing, a comparison of two small-scale platform and one large-scale platform, high-density large-scale NK cell genome editing, and last part of agenda will be generation of CAR cell and testing of the functionality of that CAR cells. So if you think about the cell therapy manufacturing, it's a very high degree of complexity as well as very labor-like and intensive. So to address, our translational and clinical cell therapy customer, Thermo Fisher dedicated to develop different CTS product, which will reduce high degree of complexity. And in coming slide, I will show you how Thermo Fisher is going to help you to, reduce the high degree of complexity of the workflow. So one of the key challenges faced by cell and gene therapy industry is poor efficiency in cell processing. A key improvement being made is shift towards closed and automated system, which can reduce the risk of contamination and error, as well as provide the ability to process multiple products in parallel in less controlled spaces. Here, I am going to discuss about the use of our CTS Rotea, CTS Electroporation system, CTS2 Cas9, and NK cell expansion medium to expand the NK cells. So this is our, the Rotea Counterflow centrifugation system. It is a closed automated cell processing system, and it's designated for cell therapy manufacturing. And this system offers exceptional cell recovery and viability, flexible input and output volume, capability making it ideal for cell separation, concentration, washing, and more. And in this workflow, we use our Rotea system to isolate the PBMC from the leuco pack with significant time saving. And in the coming slide, we will show you the data. This is our, the Genome Electroporation system. It's a closed, scalable electroporation system for GMP compliant cell therapy manufacturing. In 2016, we kicked off develop a Genome with two-fold vision. Our first vision was to enable efficient process development by allowing developer to screen a small scale with neon, and those parameters will scale to large scale genome with no sacrifice in performance. Our second vision was we needed to have an eye toward designing for use in GMP manufacturing environment. This means enable a closed system processing and GMP manufacture consumables with extensive testing aligned with regulatory compliances. And this is open platform, allow customized electroporation EP setting, and its voltage from 500 to 2500 volts, and different pulse width and pulse number. And this system is able to uh, uh, efficient with non-viral transfection, and it can be delivered different payload including RNA, DNA, and protein. This is our newly launched, is a Neon Next Electroporation system. This system is, the experimentally designed with cloud connectivity, and it has, tip technology, and this system is very efficient to transfect hard cell type, including the NK cell. In the coming slide, I will show you the data how we edit the NK cell using by this system also. This is the TrueCut Cas914, and it had high editing efficiency in different cell type, including different primary immune cell like NK cell. And this workflow we use CTS Cas9 to edit genome editing of NK cells. This is NK-Xpander Media, and our dedicated team developed NK-Xpander Media that is able to expand human NK cells without need of fetus cells. And I will share the data with you today, showing that expanded NK cell maintain their cell surface marker expression, including CD56 and CD16, and maintain a robust cytotoxicity activity. This NK expander media manufacture animal origin free at the primary level. However, supplementation with human serum is recommended for best result. NK expander is needed to meet a regulatory compliance requirement for ancillary material used in the cell and gene therapy manufacturing. So now I'm going to talk about what is the NK cell, and this is the overview of NK cell. It's a natural killer cell or innate immune effector cell that can rapidly identify and kill abnormal virally infected tumor cells. NK cell can be genetically modified to obtain effector cell with increased specificity and potency for adaptive cellular treatment of cancer patients. And CAR NK cells may represent a valuable tool complementary to CAR T cell in adaptive immunotherapy of leukemia and solid tumors. But it's hard to gene transfer in NK cell. It's very challenging. This is a NK cell therapy. The NK cell therapy can originate from multiple sources, including enrichment from the peripheral blood and cord blood, and differentiation from HSC or iPSC, and from cancer cell lines. To improve the immune cell function against the cancer or other disease, we need to engineer the NK cell. And in the previous slide, we discussed about engineering of NK cell is challenging. So you can see here, scientists try to different method to edit the NK cell, as for example, plasmid transfection, but it has limited efficiency to express transgene. Scientists also try for edit the NK cell by using the retroviral transduction, but it requires high viral titer and poses concern of insensible mutagenesis and oncogenesis. And lentiviral transduction also tried, but it has inconsistent of NK cell even at high MOI. So what the method people are trying here? So this is another method. It's called electroporation method. So electroporation method will coming the slide will show you how electroporation method will help to edit the NK cell, not even a small scale, even in the larger scale. That means where reversed and precise toolkit is urgently needed for NK cell engineering and expansion. So this is the our standard workflow, and where we are going to showing the data in the coming slide. So we isolated the NK cell from the leukophage. We isolated the PBMC from the leukophage using the CTS Rotea, then isolate the NK cell from the PBMC, and we expand that NK cell using the NK-Xpander media, having supplemented 5% serum and interleukin IL-2. And after that, once cell expanded, we edit the NK cell using Neon transfection system, Neon Next Electroporation system, and CTS like Genome system, and after that we characterize analysis with our next flow cytometry. So this is the PBMC we isolated from the Rotea system from the leukopak, and isolated PBMC phenotype was characterized here, and PBMC were stained with CD56, CD16, and CD3. You can see the left panel showing the representative cell density plot, and the right side panel bar graph represent average of like 75% T cell population and around 18 to 20% NK cell from the gated cell, and data is presented here average of 3 different donor. And we, after characterization, next we isolated the NK cell from these PBMC. So this is the the NK cells expanded. We isolated from the NK cell from the PBMC, and we use here our NK-Xpander media to expand the NK cells. On the left side panel, the graph, you can see here the NK-Xpander supplement with 5% human serum and 500 unit polymer IL-2, and feeding these cells every 2 to 3 days, beginning from the day 5. And we can see here the fold expansion is good up to 3,000 fold in 2 weeks. In the middle, the panel, we also like the NK-Xpander not maintain only the fold expansion, but it also maintain the purity. And we can see here we measure the purity of NK cell by using the antibody like CD56, and purity say more than 95%. However, we can see here the NK-Xpander media can expand the NK cell, maintain their purity, and we are interested to see the functionality. And that expanded NK cell also maintain their functionality, and that can kill the cancer cell K562. You can see this in the extreme right side panel. So now I'm going to talk here. So a small scale optimization of NK cell genome editing using the neon transfection system, and this is the workflow here. We isolate NK cell, enrich that cell, and culture for 6 days, wash and count, and then we did electroporation. And after electroporation, we culture for another 3 days. Then we start characterizing these cells by using the flow cytometry. So here we have the preset neon different program here, like program 1 to 24, and we use all program here and edit the NK cell. And here our target was AVS1, and we perform the GCDSA. And the left side panel is like the one of the gel representative figure from the gel once we perform the GCDSA. And the right side, the graph showing the quantification here. And based on, we can see the out of 24 program, like program 5 and 24, like showing best editing efficiency of NK cell. And even like some other program, I can see 1, 2, 3, 4 is also showing editing efficiency better. But based on that one, we selected top 5 program from here, and then we translate from neon to Genome system. And we can see here this top 5 protocol tested in the previous described workflow, both neon. And here we tested both neon and Genome side by side. And the summary of this protocol, we tested it you can see the like in the tabular form here. And protocol that condition are listed in the table. Any distinction between the neon and Genome protocol are also noted in the table. So the top performing protocol on both neon and Genome were, like we can see, optimization 6 and optimization 1. And these protocol result in highest, our target was like B2M just knockout, and both like optimization 1 and 6, so high knockout of B2M. Not only, we also saw the high viability of edited NK cell, and both programs show high cell viability as well as fold expansion. And that program is, you can see the tabular form in the right side, the different program have different pulse width and voltage. So based on that one, we selected only one program, is called program 5 or optimization 1, having 1700 volt, 20 millisecond, and one pulse. And in the coming slide, we are going to show the data based on that one program. We edited the NK cell in a small scale as well as in the larger scale. So first, we are going to discuss about here the small scale genome NK cell genome editing. Here we compare NK cell genome editing with 3 different electroporation systems. It's called Neon, Neon Next, and Xenon electroporation system. So this is experimental details, and our goal was, from the Neon to Neon Next and Xenon goal was, scalability from research scale like 10 microliter tips from Neon, Neon Next to developmental scale like Xenon 1 ml. And here you can see in the tabular form we have like experimental details and sample tested. So we use 50 million cells per ml, and we use Neon Next 10 microliter tips. And for the Genome, we use 1 ml single shot, and our payload was CTS Cas9, and our target custom guide RNA was B2M, and both system has same program. It's called 1700 where 20 millisecond 1 pulse as a Neon Next, and Genome system. So you can see we compare here, and the left panel in the top one is Neon, the middle one in the Neon Next, and bottom one is the Genome. So left panel showing the representative cell density plot and how we getting the cells, and the right side panel bar represent all the 3 electroporation system. So average of around 61 to 85% NK cell genome editing, and edited NK cell not only like maintain there, you can say the phenotype look like the CD56 positive is like 75 to 90% positive, and also edited NK cell maintain their viability, and viability is like 90 to 98%. And here we tested 3 different donor, is average of 3 different donor result here. However, you can see here, so NK cell therapy in the previous we saw here, we edited the NK cell in a small scale, and now we are interested for the high density or large scale NK cell genome editing. So if you think about NK cell therapy, it requires large number of NK cell per therapy dose, and multiple doses are required. And one of the key challenges faced by cell and gene therapy industry is high efficiency of NK cell genome editing on large scale. So to this end, CTS electroporation system, how they are going to help to edit different cell type on the larger scale. And the coming slide, I'm going to show the data, how CTS electroporation system is efficient to edit the larger scale NK cell genome editing. So this is the schematic, the workflow on the large scale genome editing of NK cell. Here we isolated the PBMC from the leukopipe using the Rotea, isolate the NK cell. The same we expand the NK cell in the NK-Xpander media, supplement with human serum and IL-2, and then on the day 6, we read the NK cell here Neon and compare the Genome system, and after 72 hours, we characterize the characterization and analysis of the edited NK cells. Our goal was to understand the impact of performing high density electroporation NK cell by comparing the electroporating at the different cell density 50 million to 100 million cells per ml. And we can see the experimental details here. We tested here 2 different donors, and Neon system used 50 million cells per ml and 100 microliter tips, and Genome system we used 50 million cells per ml, 1 ml single shot, and 100 million cells per ml, 1 ml single shot, and we use payload is TrueCut Cas9 protein, and our target was B2M, and we use the genome editing CTS buffer, and both Neon and the Genome we use the same electroporation parameter. It was like 1700 volt, 20 millisecond, and 1 pulse. So there you can see here the left panel showing the representative cell density and how we get it. The top one is the Neon, middle one the Genome and the 50 million cells, and the bottom one is also Genome 100 million cells. And in the right side panel, the bar graph represented Genome electroporation system. So you can see the editing efficiency of like is showing more than 85% knockout in high density. Not only the 85% knockout, but it also maintained the phenotype. You can see the like CD56, so more than 90% positive in edited NK cell, and as well as it's also maintained the viability. We can observe here more than 95 to 97% edited NK cell is viable. So our summary is like the PBMC we isolated using the Rotea system, and pre-isolation average, you can see the CD56 around 14%. And NK cell we isolated from the 3 different donor in the PBMC, it saw 90 to 95% CD56 positive. And NK-Xpander media, we use, and its average is around 2,000 to 3,000 fold expansion. And Neon Next system shows similar NK cell genome editing efficiency, and CTS Genome system so around 85% knockout efficiency in average of 3 donors. So in the previously we can see here our electroporation system is able to edit a large scale in the knockout. Now we are interested about the knocking. So in the coming slide, we are going to talk about the generation of the CAR NK. Especially we are going to talk about major 3 CAR NK cells. So the CAR NK, this is like a little background of the CAR NK. The CAR NK is, the chimeric antigen receptor, an artificially modified fusion protein that consists of extracellular domain fused to intracellular domain, and primary NK cells engineered to express CAR have potential advantage over CAR T cell, and CAR have been generated variety of different, the target antigen, including like CD19, CD22, so on, and mesothelians. I highlighted mesothelium. I'm going to discuss about the mesothelium CAR here. And different methods have been used to introduce CAR into the NK cells, like including viral transduction and electroporation method. In the coming slide, we'll discuss about the viral transduction method as well as electroporation method to generate the CAR NK. And the CAR NK cell can be validated using different methods, including flow cytometry, cytokine production, and endpoint cytotoxicity. So first, we here try for the viral transduction here. In the top panel, we have like control cells, like just NK cells with no any virus here. So most important here, we use here AV6 having major 3 single strand donor DNA. The top panel is like one NK cell without any virus, and we didn't see any transduction of AV6. Then here we use high like 200K MY of AV6 having major 3, and we incubated the NK cell with this high MY of AV6 major 3 for 3 days and 7 days, and we performed the flow cytometry either the day 3 or day 7. We didn't see any like viral transduction induce the CAR NK. That means like the viral transduction alone is ineffective to deliver the the gene of interest CAR in the NK cells. So now what are the different methods we can think? So based on that one, we are thinking we can do the electroporation method. So this is the the overall workflow here. So we isolate the PBMC again from the Rotea system and isolate the NK cells from the PBMC, expand the NK cells using our NK-Xpander media, and here we use Cas9 and RNP mix, where we try to the AVS side, AVS1 knockout by using the electroporation system, and in that side we trying to knock in having the AAV6 major 3 homologous arm. So once we knock out at the same region, we're trying to knock in, and then after knocking, the cell is expanded for the 6 to 21 days, and over the time period we measure the NK phenotype, percentage viability of the cells, and the endpoint of we measure the cytotoxicity of CAR NK. So this is the the experimental and the sample details here. So here we tested 3 different donors, and concentration of cell during electroporation neon is 50 million cells per ml in 100 microliter tips, and we use Genome system 50 million cells per ml, 1 ml single shot, and our payload is a true cut Cas9 protein, and we have ABS1 guided a knockout and ABS6 meso 3 we give to knock in here, and we use our CTS genome editing buffer, and both electroporation system neon and the Xenon had the same parameter 1700 volt 20 millisecond and 1 pulse, and our goal was to understand the role of the electroporation or generation of CAR NK cells. So here we compare with two different electroporation systems. The top one is the just control cells, like one of the representative I'm saying the how we getting the cells here. So we use the NK cells, only 100 kMOI, AV6 having meso 3, and we incubated for the different time point. We measure the different point of, and we didn't see the virus has any, transduction or generation of CAR NK. The middle one, we edit the NK cell, we knock out the ABS1 region with the neon, and the same region we try to knock in with the AV6 meso 3, and we can see the neon show around 26% CAR positive cells. However, the same way the genome showing around 37% CAR positive in NK cells. The right side panel, we measure the expression of meso 3 like over the different time point, and we can see the genome system is average of like around 32%. However, the neon system showing only 18%. And most importantly, we can observe here, it vary from like high, we can see the donor to donor variation is very high here. And then we, in the top panel, we also measure the total, viable edited CAR NK, how much we have. And over the time, we can see the CAR NK viable edited cell expansion is high. And the left panel, in the bottom one, we measure the viability after electroporation, and both like NK cell or CAR NK like maintain their viability. And the extreme bottom one, right panel, however we can see the like NK cell is viable, but we also measure the immunophenotyping. And the right panel, we can see the either NK cell or CAR NK maintain their phenotype, and you can see the CD56 is positive is more than 90 to 95% here. However, we can see we develop the CAR NK, and we can see the over the time its expression is increased. We can see the viability, the cells have more viable and they maintain their phenotype. The endpoint, we are interested to see the functionality of the developed CAR NK. So here we take the SKOV3 GFP cell line, cancer cell line, and we incubated effector versus target ratio was 10:1, and bottom like maximum and minimum is 0.6 to 5:1. And we can see here the one of the representative image from the microscope, the left side panel. Here we incubated effector and target cell only for 6 hours. And after 6 hours, we can see even the image showing the meso CAR NK has like more killing efficiency as compared to normal NK cell. And most accurate, we can see the right side panel, we measure by using the flow cytometry, and we can observe that even the 2.5:1 ratio CAR NK has high, killing efficiency, even so more than 40% killing efficiency of their target cell, and 5:1 is showing more than 80% killing efficiency against their target cell SKOV3. So we can summarize here, the CTS genome system successfully used for gene editing, and we use our CTS, the NK-Xpander media to expand the CAR NK. The CAR NK not maintain the only phenotype, they also maintain their the functionality, and CAR NK cells were able to kill around 80% of target cell within 6 hours of co-incubation. So in this way, our CTS Rotea system and genome system are powerful modular tool in the quest towards creating a closed cell therapy manufacturing process by providing superior performance and helping reduce contamination in cell therapy manufacturing workflow. Thank you.
Well, thank you very much indeed, Deepak, for a great presentation there. And we've had lots of questions start coming in already from the audience, so I think we should start our Q&A session. And so a few questions have come in regarding electroporation. So I wonder if we could start there. And could you tell me a little bit more about how you would optimize electroporation conditions for different cell types and payloads? And this ties in as well with a question from the audience member regarding, are the electroporation parameters for the Neon and the Xenon always identical for different cargoes and cell types? So perhaps we can combine that into the one.
Okay. Thank you so much. So for the first, we optimize like in a smaller scale, we use the Neon system. And during optimization, we have to like focus on electroporation protocol, especially for voltage. And as we mentioned, how different voltage behave in the different types. And then some variables to be considered during optimization, like electroporation program, cell density, payload concentration, and starting material. And once you electroporate, the most important is also how you are culturing the cells. So these are like the we can consider during optimization protocol for any cell editing.
That's great. Thank you very much indeed. And another question that's come in from the audience, at what day post-activation, post-seeding, do you electroporate or transduce with AAV the NK cells?
Okay, thank you. So we not activated, we isolated the NK cell culture for the 6 days. And on day 6, we start editing the NK cell. And if you see in the workflow, we clearly mentioned that on the day 6, we use the electroporate, just knock out one of the region called ABS1 region, and after 30 minutes, we gave the virus, and then after that we culture for like 6 to 21 days. This is the way we did the knock-in one.
Perfect. Thank you. And our next question, could you provide more details about how Rotea helps prepare cells for Xenon electroporation?
Oh, thank you so much. So, the electroporation requires removing of the cell from the media, washing them, concentrating them, and then resuspending them in electroporation buffer, like very high density, like 20 to 100 million cells per ml. And the Rotea is uniquely suitable to output the small volume required for the electroporation and complete the process in closed or called semi-automated manner. So this way, Rotea helps to prepare the cell for Genome electroporation.
Fantastic, thank you. Another question from the audience. What is the AAV MOI used after electroporation?
Sorry, what's your question?
What is the AAV MOI used after electroporation?
Okay, yeah. So I use the 100K MOI. So what we did, first we optimized in different MOI, and we found like 100K MOI give the best result. So that's why we use here only 100K MOI.
Perfect, thank you. And another question can you use the Rotea to process expanded NK cells?
Oh, thank you so much. Yes, definitely we can use. So Rotea is capable of performing wash and concentrate on NK cell, like similar like the T cell. We have like slight setting adjustment, and based on we have like several app note on the Rotea website, and they're detailing the capabilities. So if you go to the website, we can find like all app note regarding the T cell, but we have like slightly different setting for the Rotea process expanded NK cell also.
Great, thank you. And our next question, what is the maximum number of cells that can be washed and concentrated using the CTS Rotea system?
Well, that's a great question. So in the Genome system, you are in the Genome system, right?
Sorry, the CTS Rotea system.
Oh, CTS, sorry, thank you.
No worries.
So Rotea has the capacity to process up to like 5 billion cells, although this number may vary depending on the type of cells. And each chamber, if you see the Rotea load, can handle the maximum number. However, for the larger batches, Rotea can come refill multiple times. And moreover, by incorporating multiple loop in the protocol, Rotea is technically capable of process like a maximum like in case of like T cell, we have like up to 5 million. The and the NK cell we have around 4 million cells.
Great, thank you very much indeed. Another question in from the audience. Have you tested any other viral vectors within your system, such as Lenti or Retro?
Yeah, in the coming we are now in the currently we are doing right now. And hope very soon the result will come out.
Oh, excellent. Yeah. So watch this space. Next webinar.
Yeah, maybe. Yeah.
Excellent. And another question in from the audience. So how did you detect the CAR expression in NK cells by flow cytometry?
So yes, so in that workflow, you can see here, so we have the tag, it's called V5 tag there. And once we incorporate it and over the time flow, and we just do the flow cytometry to measure the percentage of CAR NK by using the just V5 antibody. And this way, we measure the percentage of CAR NK, how much CAR NK is there.
Great, thank you. And what is the maximum volume and number of cells that can be electroporated in a single run on the Xenon system?
Oh, that's a great question. Thank you. So, well, for the, if you see the Xenon has like multiple sort and single sort. For the single sort, like 1 ml, like in the my workflow, we use like 100 million cells, but it can be electroporate higher cell density safely, like up to 300 million cells per ml, but optimization should be done. For the multiple sort, like in 25 ml, like it's able to like 2.5 billion cells. They can electroporate the Genome system.
Fantastic. And how long is a typical Xenon run?
In case of like 25 ml multi-sort, it took like 20 to 25 minutes to complete once once we uploaded all consumables, everything. And for single sort is very quick, like 2 to 3 minutes maximum.
Oh, wow. Yeah. Great, thank you. Okay, and another question for you. So what other cell types have you shown to be successfully edited using the Xenon?
Oh, that's great. So internally, we have tested many cell types, like we tested activated T cells, resting T cells, iPSC, HSC. Like in this webinar, we presented like NK cells. Then we have tested like monocyte, Jurkat, so many like this.
Wow.
This has been tested, yeah.
Excellent. And another question. Can CTS NK-Xpander be used to expand pluripotent stem cell-derived NK cells in a feeder-free system?
That's a great question. Yes, we have worked with the one of our collaborator to test the iPSC-derived NK cell expansion with our NK-Xpander, and their results showed like NK-Xpander resulted in better INK fold expression in the feeder-based and feeder-free system compared to the classical media-based system. And even our like NK-Xpander media is like feeder-free. And one of the like Dr. Dean Lin, he presented in the webinar and discussing his work with the NK cell. And you can find like he used the like optimization of NK cell culture media with our NK-Xpander, and as well as K562 feeder cells. And he found like NK-Xpander like free like if you don't use any feeder cells, it expands very well, and it's able to kill the target cell also. And for more detail, it's available at the thermofisher.com/NK cell therapy. You can find the more detail there.
That's great, thank you. Another question in from the audience. So are the cytokine concentrations altered from normal culture conditions at any point before, during, or after electroporation?
No, so we maintain the same, like, the IL-2 concentration here from like pre and post. And we didn't find like any alter of like the NK cell phenotype or in the either phenotype or functionality.
Great, thank you. And another question from the audience. Can you cryopreserve the NK cells after expansion?
That's great. Currently, we are working on that one. So I can see the many paper they discuss about cryopreservation for the NK. And the problem is the once we preserve, the problem is the once we thaw, the expression of CD16 down, and we are currently working on that one.
Thank you for sharing. Great. And then another question that's come in. How much fold expansion growth is observed in edited NK CAR cells?
Okay, so that will go, if I correctly remember, it go up to 60-fold higher in the over the time.
Fantastic. Thank you. And then do I need to be worried about electroporation temperatures affecting cell health?
That's great. Yes, so for the, so we have like program safeguard onto the software. So for which this kind of like occurring. So we don't have like worry about that one.
Excellent. And then a few more questions that are coming from the audience. So why do you transfect after 6 days? Is there a difference in electroporation performance over the expansion period?
Yeah, that's a great question. So we optimize this one. So we try to edit the NK cell on different days. And we found like day 6 is the best day for editing. And if you see the even on the day 6, we are thinking like the NK cell have like, cell cycle, cell maturation is like more accurate day on that day 6. So based on that one, we are thinking day 6 is the best day. So that's why we choose the day 6 for the editing one.
Yeah, that makes sense. Great. Thank you. And another question from the audience. What is the minimum volume that you can use to resuspend cells in the Rotea?
It should be like in the 5 ml.
Oh, sorry, I didn't catch that.
5 ml.
5 ml.
Yes.
Excellent, thank you. Yeah. And then our next question, does electroporation efficiency change depending on the day of expansion of the NK cells? Which I think you...
Yes, we just a few minutes ago we discussed about that. Exactly. Yes. Yeah. So over the time, and we can see the like after like day 7 or day 10, the editing efficiency decrease.
Great. And another couple of questions in from the audience. So what platform did you use to expand your cells with NK-Xpander Medium?
So we use here our G-Rex. We have the G-Rex culture media, platform. And even we have like the normal plate also, but G-Rex give the like best result.
Great, thank you. And then I've got another question. Can you expand NK cells from PBMCs without enrichment?
We never try that one. Because the PBMC has like the lots of T cell, and definitely is a great question. We can try that one, but we never try that one.
Excellent. Oh, well, thank you so much, Deepak, for answering all of our questions there. You did a fantastic job. And thank you, as always, to our audience for your great questions. If there were any that we couldn't answer directly today, please rest assured they'll be passed on to the Thermo Fisher team and they will reach out to you directly. But I just need to say a big thank you again to Deepak for a really great presentation today. No, it's been a pleasure speaking with you. And as I said, thank you to our audience, as always, for joining us today. The on-demand will be available tomorrow, so do look out for an email from us in your inbox and do feel free to circulate it. And then we hope to see you again for our next webinar. Thank you again for joining us, and thank you, Deepak. It was great chatting with you today.
Yeah, bye.